I am T. Sandhya, Department of Zoology, Andhra Loyola College. Dear students, in this video we are going to learn about one of the most important mechanisms happens in the living systems or replication of DNA. Before learning the mechanism of replication of DNA, first let us know what is DNA, what is replication and what is its significance. DNA is one of the biomacromolecules in the living systems or living organisms. It is the genetic material that carries the information about the function, growth and repair of everything in the organisms. It is the material of inheritance also. If we look into the structure of DNA, it consists of two polynucleotide chains that are directed from the 5 prime end to 3 prime end, and they are held in opposite directions. That is why we describe it as anti-parallel to each other, and these two strands are held together by hydrogen bonds between complementary bases. Now, what is called replication? Replication is the mechanism of production of duplicate copies or replicas. So, replication of DNA is production of replicas of DNA molecules from the original parental DNA molecule. If we look into this slide, the parental DNA molecule is represented with two template molecules that are wind around each other in a helical manner. The complementary bases, for example, adenine in one strand is held with thymine in opposite strand with 2 hydrogen bonds and vice versa, and guanine in one strand held with 3 hydrogen bonds with cytosine in opposite strand and vice versa. This complementarity is the principle in the process of replication mechanism. In this slide, we can see one parental double stranded DNA molecule got separated, and the separated strands act as template molecules to synthesize new copies or new complementary strands, and that results the formation or production of the two daughter double stranded DNA molecules. In each daughter DNA molecule, we can see one strand is old parental strand, that is the template, and the another strand, its complementary strand, is new strand that is being synthesized. That is why it is called as semi conservative mode of replication. So, replication is nothing but the information present in one of the strand is being copied very much precisely, and this happens during the S phase, that is the synthesis phase of the interphase in the cell cycle. We have discussed about this in the previous classes. If we take the example of human beings, each human cell consists of 3 billion base pairs of DNA pairs. The two nucleotide strands, that is the polynucleotide chains, as we discussed earlier, they are held together by the hydrogen bonds between the complementary bases, that is A base pairs with the thymine with the 2 hydrogen bonds, and C base pairs with, that is a cytosine with guanine with 3 hydrogen bonds and vice versa. So, the semi conservative mode of replication results the production of one old strand and one new strand in the newly produced double stranded DNA molecule. And coming to its significance, the very first one is it is the basis for inheritance of characters and its transmission from one generation to another generation. No other molecule in these biomacromolecules classes possess this particular feature of replication. So, because of this process of replication only, the genetic information would be carried on to the from one generation to the another generation. So, we look into the mechanism. There are 3 steps mainly involved in replication. One is initiation, second step is elongation, and third step is termination. So, the point where exactly the replication begins is called as origin of replication or replication origin. This region is generally rich in AT bases, that is adenine and thymine base pairs. So, this particular region would be identified by certain proteins called as initiator proteins. They will identify this origin of replication and will go and bind there. Then it is followed by the enzyme helicase, which goes to the particular point of origin of replication and binds to the initiator proteins to begin the replication. After this, the primase enzyme, that is RNA primase, will go and bind to this complex to begin the DNA polymerization, that is replication. So, RNA primase synthesizes the RNA primer. So, initiation involves the origin of replication, that is exactly where the replication begins. In higher diploid organisms, there are many points of origin of replication, and so this requires the separation or unwinding of the 2 complementary strands that are held together by hydrogen bonds. So, this unwinding is brought by the helicase enzyme. So, the unwinding results the formation of a fork-like structure. So, this fork-like structure is called as replication fork. So, let us see what are all the enzymes involved in this initial step, that is initiation of replication, initiator proteins, helicase enzyme and single strand binding proteins, which prevents the reunion of the complementary strands till the synthesis is being completed. And another important enzyme, that is DNA polymerase enzyme. DNA polymerase enzyme is the enzyme that synthesizes the complementary strand against the template strand. So, have you look into this slide. So, where the 2 strands got separated and a fork-like structure is formed, that is called as replication fork. So, DNA polymerase enzyme begins the synthesis after a small oligonucleotide, that is RNA primer, being synthesized by the RNA primase enzyme, because DNA polymerase enzyme cannot initiate the replication. That is why RNA primase begins the replication and it synthesizes the small strand of RNA primer. And this synthesis is continuing in the direction of 5' end to 3' end, because the DNA polymerase enzyme always requires a free OH group at the 3' end of the molecule. So, this is another slide that gives us an idea, overall idea regarding the DNA replication. If we look into this slide, one strand, the new strand is being synthesizing continuously, whereas the other strand is synthesizing in a small fragment like structures. The strand that is being synthesized continuously against the 3' to 5' template strand of the parental DNA is called as leading strand, because this is providing the free 3' OH end continuously. But the another parental strand, that is in the 3' to 5' mentioned, is being copied in fragment forms. These fragments are called as Okazaki fragments, and these fragments are being again synthesizing in the same direction, that is 5' to 3' direction only. So, let us see the elongation. So, the template strand dictates what nucleotide bases should come and join or add to the free 3 prime OH end. So, one of the strand, as we have seen in the previous slide, is read in 3 prime to 5 prime direction, and the synthesis is always from the 5 prime end to 3 prime direction, and new nucleotides would be added one after another according to the guidance or information that is present in the template to the free 3 prime OH end of the previous nucleotide base. So, there are 4 nucleotide bases, deoxyribonucleotide bases, that is DGTP, DCTP, DATP and DTTP in the cytoplasm. They are involved in this synthesis. So, the leading strand synthesis is on 3 prime to 5 prime template strand, that is the parental template strand, whereas the lagging strand is synthesized against the template strand which is in the 5 prime to 3 prime direction. Whatever may be the direction of the template or parental strand, the new strand would always be synthesized only in the direction of 5 prime to 3 prime direction only, because the addition of nucleotide always requires a free 3 prime OH end, that is a free OH end at the third carbon of the deoxyribose sugar molecule. The RNA primers are added to the newly exposed bases in the lagging strand. So, many RNA primases would be involved in synthesis of many RNA primers, as the separation of the two parental strands is being continuing. DNA synthesis that is happening in fragments is they are called, fragments are called as Okazaki fragments. They are also in the direction of the 5 prime to 3 prime direction. And the last thing is termination. So, no more synthesis of DNA takes place after this reaching this terminal point or end point. So, newly synthesized synthesizing strands would be stopped and it would be stabilized. So, here the ligase enzymes will join the Okazaki fragments to complete the lagging strand also. So, the purpose of DNA replication is to create 2 daughter DNA molecules that are identical to the double stranded parental DNA molecule.